Do we have some students from ULB now? Two? Do we have students from ULB, please? Oh, ils sont timides, hein? So, ladies and gentlemen, please welcome now a researcher at the Laboratory of Histology, Neuroanatomy and Neuropathology at ULB, Professor Karelle Leroy. Researcher at the Laboratory of Histology,
I want first to thank the organizer to let me the opportunity to show you the results and the works that we are doing in the lab, and of course in other labs. I will present you the cellular and mouse models of Alzheimer's disease, some of them, of course, because there are numerous models of Alzheimer's disease. Just next slide. In Alzheimer's disease, we have two neuropathological lesions that we can find in the brain. The first one is called the neurofibrillary tangles. It's accumulation in the cell bodies of neurons. We see here the brown color that shows the pathology here. And the second lesion is called the senile plaques. The senile plaques are composed of an extracellular deposit of amyloid peptide in the brain. This amyloid peptide here is also colored in brown. Next slide. Thus we have seen that in this plaque we have the amyloid peptide. This amyloid peptide comes from the cleavage of a precursor called amyloid precursor protein. I will call it APP in the future. This APP protein can be cleaved by two pathways here, two pathways. The first one is the non-amyloidogenic pathway. You must see that it's not generating amyloid peptide. Why is it not generating amyloid peptide? Because the APP here is cleaved inside the sequence of A beta that is colored in yellow-orange. This cleavage is due to the alpha secretase activity. In the amyloidogenic pathway, we have two successive cleavages. The first one and the second one will generate the amyloid peptide. This amyloid peptide accumulates in the extracellular domain or in the brain and accumulates to form the amyloid plaques. This APP here, I will show you later, but this APP can have mutation in the sequence leading to the accumulation of A beta in AD brain. Next slide. For the second lesion, the neurofibrillary tangles, we have thus an accumulation in the cell bodies here in brown. This accumulation, when we looked at these lesions by electron microscopy, we can observe paired helical filaments. Why paired helical filaments? Because there are twisted filaments that are present in the brain. These filaments are composed of two proteins. Why is it interesting to study this lesion? It's directly correlated with the cognitive deficits. Here it's a curve showing the abundance of the lesion in the cortex and the cognitive score of the patients. And we can see that there is a strong correlation. More you have tangles, more deficit you have. Next slide. But what is tau? Tau is a member of a family of protein. It's the microtubule-associated protein. These proteins bind to the microtubules, and they stabilize the microtubules. Next, animation, yes. This protein contains, in fact, in the C-terminal part of the protein, some binding domains, so the microtubule binding domains. It's also called the semi-repetitive domains. It's with this domain that the protein is able to bind to the microtubule. Next. In cells, when we express two proteins coupled to GFP, GFP is for green fluorescent protein, we can see all the network the network, microtubule network in the cells, is due to the fact that the tau protein that is fluorescent binds directly the microtubules and label the microtubules due to this binding. Next slide. The gene for tau is located on the chromosome 17 in the human brain. Next. And it's transcribed in 6 isoforms. Next. Thus here we have the 6 isoforms that we can find in the human brain, human adult brain. These isoforms differ by the absence or the presence of 1 or 2 inserts in the N-terminal part of the protein. That's why it's called 0N, 1N, or 2N. And by the presence of 3 or 4 microtubule binding domain. As it's repetitive domain, we call them 3R or 4R isoforms. And thus it gives rise to 6 different isoforms present in the adult human brain. It's particularly important to know that it's in adult brain because in embryonic brain, we can find only the shortest isoform, the isoform without insert in the terminal part of the protein, and 3 repetitive domain. Next slide. When we look to tau proteins in the neurons in physiological conditions, these proteins are located around the microtubule network, and we can find these tau proteins in the axons, mainly in the axons. When we do labeling on tissues, on brain tissues, we can see dots like this here. It's due to the fact that the proteins is located in the axons. Next. This binding is regulated by the activity of kinases and phosphatases. When tau proteins are phosphorylated, they detach from the microtubules, and the microtubules are not more stabilized, they are depolymerized in this condition. It's particularly important because these microtubules play an important role in the exoplasmic flow in the axons. Next. But what happens in Alzheimer's disease? In Alzheimer's disease, we have a big change. There is an imbalance between the activity of kinases and phosphatases, and thus the phosphorylation of tau is highly increased in Alzheimer's disease. Next. This highly phosphorylated tau is not more able to bind to microtubules, and thus the localization of tau change inside the cell bodies of neurons. And thus we can see here that tau proteins accumulate in the cell bodies of neurons and are not present anymore in the axons. And the localization seems to be insoluble because we see aggregates in the cell bodies of neurons. Next slide. When we look to the steps for the formation of the pathology, we can see the first step is the phosphorylation. The second step is the aggregation of tau proteins that finally form paired helical filaments. These paired helical filaments accumulate in the cell bodies of neurons to form the tangles. These tangles contain the 6 isoforms of tau that we can find in the adult human brain. Next. The kinases are thus responsible of this abnormal phosphorylation, and there is a link between the amyloid peptide that is in the over lesion, the senile plaques, and the tau, because if you if you check the composition of the senile plaques, we can see that it's amyloid peptide, and it's known that the amyloid peptide is able to activate the kinases responsible of the abnormal phosphorylation of tau. Thus there is a link between the 2 lesions that we can find in Alzheimer's disease. Next. And thus the pathology have different stages. The pre-tangle stages, the tangle stages, and the ghost tangle stages. It depends of the epitopes that you can find. Here I show you the phosphorylation. The epitopes are not appearing at the same moment. Some are more early than the others. Next. And the aggregation appear only on the stage 2 and the stage 3. Thus, we can see that at the pre-tangle stage, we have phosphorylation of tau but no aggregates. At the stage 2, thus the mature tangles, we can find phosphorylation and aggregation. And at the stage 3, we do not find any more phosphorylation, but we can detect aggregation. It's due to the fact that these neurons are dead, and thus we have a ghost tangles. Next. And thus the stage 1 and 2 are found inside the cell. The stage 3 are extracellular. Next. Yes, here we do not see clearly the pictures, but here in these pictures that we have no aggregates, but we can detect phosphorylated tau in the cell bodies. It's the stage 1, thus with the pretangles. In the stage 2, we can detect pathological tau in the form of aggregates, and phosphorylated tau in red. In the stage 3, we can find aggregates of tau, but we cannot detect anymore the phosphorylated tau. Thus the neurons here, if you can check, you do not detect anymore a nucleus, and thus the cells are dead. There is no more nucleus in the cells. Next. The difficulty now is to modelize the tangles. You will see that for the amyloid plaques it's quite easy to modelize, but tangles is very difficult to modelize because these models need to have these different characteristics. Thus an abnormal localization of tau, an abnormal phosphorylation of tau, the presence of filaments, and the presence of aggregates inside the cells. Next. I will show you just 2 models of cells. Why? Because we do not succeed to form pathology in cells. It's impossible. I just show you the first one is the primary cultures of neurons. This is really neurons in culture. We take embryos from mice. We dissect the embryos. We take the brain. We remove the meninges, and we put them in cultures. After a few days, we can see cells with processes. They have axons, they have dendrites, we are able to do synapses, but we are not able to see any taupathology in these cells. We wonder if it's not due to the fact that the embryos are expressing only the shortest isoform of tau. If you remember my illustration with the isoforms, we have seen that there is only the shortest isoform that is expressed, and in human we have the 6 isoforms. And thus we were wondering, okay, perhaps it's due to the fact that we do not have the 6 isoforms. This next slide. Thus we have used another model. It's a model in human. Thus we take healthy or AD fibroblasts. These fibroblasts are put in culture, and with viruses we express miRNAs. This miRNAs is short RNA sequence that can regulate plenty of genes in the cells, and it regulates the genes to push the cells in the neuronal lineage. Here we can see cells. We have neurons with dendrites, axons, synapses. They express the 6 isoforms of tau, like in the human adult brain. They are human cells. But again, we do not generate any taupathology. This is not sufficient to generate a model in cells. And thus as we want to have a model, we will look at the models in mice. This next slide. But to generate a model of mice, we need to know how the tangles are generated in the humans. And in humans, we have, in fact, 2 kinds of AD cases. We have the familial cases and the sporadic cases. When we look to the graph, most of them are sporadic cases. This is 95% of the cases are sporadic. Thus, we do not know exactly the cause of the disease. We just know that the aging is an important factor of risk to develop AD. The sporadic cases are affected after 65 years old. Thus, it's late onset. Compared to the early cases, here, it's less than 65, it's around 40-45 years. The familial cases are due to mutations in APP, presenilin 1 and presenilin 2. I will explain you later what is it. Thus first, we will focus on aging. We can change. The aging is the main factor risk. Thus we look to the very aged mice, what happens, and again, we do not detect any taupathology. Thus the aging in mice is not sufficient to generate a taupathology. Next slide. Yes, next. Okay. In the familial cases, as I explained, we have mutation in APP, that's the precursor of the peptide amyloid, the presenilin 1 and the presenilin 2. Just to remind, next, we have yes, the mutation are found in this precursor directly, or in presenilins. The presenilins are members of the gamma secretase activity that cleaves the precursor to generate A beta. When the human express this mutation, they increase the production of A beta and formation of amyloid plaque. And they have also tangles. They have both lesions. Thus we wonder if we express APP or presenilin 1 with mutations found in Alzheimer's disease, normally we are able to generate amyloid plaques but also tangles. And thus next, we have studied a model of mice that express the human APP with mutation found in Alzheimer's disease and presenilin 1 with mutation also found in different familial cases. These mice express only this mutation in the neurons to see if we are able to generate the pathology found in AD. This next slide. Thus we were very happy because in these mice we generate amyloid plaques. They are here shown in red. We can see dystrophic neurites. This is processes from neurons that are are suffering due to the toxicity of amyloid peptide. But again, we are not able to generate tangles. Next, yes. Thus again, there is no tangles at all in these mice. The amyloid plaques in mice are not sufficient to generate the tangles. And thus it takes decades because we generate plenty of mice, different models, and never we obtain tangles. I will show you another model. Next, yes. Why we are using another model? Because in mice, what we have seen, the isoforms of tau changes with the time. During the postnatal delay, we see that the 4R isoform increased and the 3R isoform disappeared. And again, in neurons, in the Alzheimer's disease, we have seen that these peridelical filaments are composed of the 6 tau isoforms. And thus we can wonder if the mice are not able to generate tangles because they do not express the 6 isoforms of tau. This next slide. And thus we have used another model, a human wild-type tau, we will call it H2 model. These mice express the 6 isoforms of tau like in human adult brain. And thus, next slide. Again, no chance, we do not generate tangles. Thus here we see the total tau, thus we can detect tau in the cell bodies, but we do not detect phosphorylation or aggregation of tau in these models. Next. And thus, during decades, we were completely blocked. We have no model to generate tau tangles. When we look to the human cases, in fact, we can have an evolution of the disease and in 6 stages. It's due to the fact that the formation of tangles appears always in the same brain regions and in the same order during the evolution of the disease. It starts in the entorhinal cortex and the hippocampus, and after it develops in all the neocortex in the later stages. The symptoms appeared in the stage 5 and 6. It's very in the latest phase of the AD symptoms are here. And thus we wonder why these regions are affected always in the same order. And when we look to these brain regions, in fact, they are synaptically connected. And we can wonder if the pathology is not propagating between neurons. This next slide. Thus, we have a hypothesis. The pathological tau is secreted by the cells, enters in a healthy neurons, and the normal tau protein that is expressed in the cells is seeded, recruited by the pathological tau to induce a new pathological form of tau, like a prion, in fact, thus suggesting a role like the prion. Next slide. To test this hypothesis, we want to inject pathological tau from human brain in the brain of mice to see if it acts as a prion. Next slide. And thus in the lab, we are able to isolate pathological tau from human brain. Thus we take brains from AD cases or control cases, thus without dementia. We homogenize the brain. We add a product called sarcosyl. Sarcosyl is a treatment to solubilize all the proteins except the pathological tau, because we have big aggregates that are resistant to the detergent. After we centrifuge at very high speed, we obtain a pellet. And when we look to the content of this pellet, yes. When we look to the content of this pellet by electron microscopy, we can see filaments like in AD. Thus we obtain the AD PHF tau from in the brain of human brains. When we check the expression, we see that we express tau. This sarcosyl fraction contains tau that are highly phosphorylated. And when we look to the non-dementia cases, there is nothing. It's a proof that we are able to isolate only the pathological tau and not the physiological tau. Okay, next slide. Thus we take this fraction containing pathological tau, we inject them in the brain. After 4 days, we checked and we are able to detect the injected material. 7 days after the injection, we are not able to detect anything in the brain. And 3 months later, with the mice injected with the sarcosyl fraction from Alzheimer's cases, we observe accumulation of tau, here in brown color. These accumulations are highly phosphorylated, highly aggregated, and recapitulates all the characteristics like in AD, like in Alzheimer's disease. Thus we reproduce completely the pathology observed in human. When we look to the cases without any tau pathology, we do not observe any pathology in the brain. When we inject the same cases here, but we remove pathological tau from the fraction, we do not observe anything in the brain. Thus, it's a proof that the pathological tau in the sarcosyl fraction from Alzheimer's cases is able to seed and recruit normal tau like a prion. Next slide. After, we were wondering what will be the effect of amyloid in this case, in the propagation of tau. Thus, we have injected mice expressing the mutant APP and presenilin that develop plaques with this sarcosyl fraction. Here I compare the wild-type mice that we have just seen. This wild-type mice is mice without any modification, and mice with mutation in APP or presenilin. All of them are injected with Alzheimer's cases. We observe this accumulation of tau in the brain, in the wild-type mice, and we observed a big increase in tau pathology in mice with amyloid plaques. Thus we can show that the propagation is increased with the presence of amyloid. When we use exactly the same mice, but injected with a case without any dementia, thus any tau pathology, we do not observe any pathology. This is a proof again that the injection of pathological tau induces a pathology, but is increased when we have amyloid peptide. Next slide. When we look to the distribution of the lesions in the brain, here is the injection point. This is where we injected the pathological tau. We see that in the more anterior and the more posterior part of the brain, we can still detect pathology in the FED mice, but no, not in the wild-type mice. Thus, we increase the pathology inside the brain, but also the propagation inside the brain in the anterior-posterior axis. After that, we looked at the H2 model. In these mice, we express the 6 isoforms of tau to see if we have observed the difference between the wild-type mice and these mice expressing human tau. Next slide. Thus we have injected AD-sarcosyl fraction again, and the difference here, we do not observe only axons, we observe also cell bodies, like in human. Thus we reproduce exactly the same pathology that in human cases. Thus we have accumulation in the cell body, we have a phosphorylation of tau, aggregation of tau, all the epitopes are present. Next, when we inject sarcosyl fraction from a non-demented patient, we do not observe anything. There is no pathology that is induced in this model. And next, and again, if we remove pathological tau from the sarcosyl fraction, we do not observe any pathology in the brain. Next. Now, we wonder if the pathology can be propagated between neurons due to the synapse. And thus, we inject mice in the dentate gyrus. Here is the hippocampus. The hippocampus here is composed of Hammond's horn and the dentate gyrus. The neurons in the dentate gyrus are connected to the sector 1 of the Hammond's horn. They are connected to the sector 2 of the Hammond's horn, and after to the entorhinal cortex. That gives again a connection to the dentate gyrus. And thus we will inject in the dentate gyrus the pathological tau to see if the different connections are also propagating the pathology. Next. Thus here you see the hippocampus, thus the Hammond's horn, the dentate gyrus. The neurons here are stained in purple. We inject here in the dentate gyrus, and when we look to the dentate gyrus, we see neurons affected with the tangles. Next. When we look to the first connection, we see an accumulation of tau, but when we looked at the next connection here, we see again pathology in the neurons. It indicates that it propagates between connected, synaptically connected regions. After, we want to know, yes, come back. We want to know where will go this pathology. This next slide. We have seen that the injected site is affected in the dentate gyrus, in the Hammond's horn, but surprisingly, we observed also a pathology in the dentate gyrus and the hippocampus in the non-injected side, where we do not inject anything. And in fact, it's due to the propagation of the pathology between the two hemispheres of the brain. Next. We have seen that the Hammond's horn is connected to the entorhinal cortex. The entorhinal cortex is very posterior in the brain. The position is is at the end of the brain. And we can see that in the entorhinal cortex, we have a tau pathology in the brain. We have studied the effect of aging in this propagation. Thus, we have injected mice at 6 months and other mice at 12 months of age. And we have observed a strong increase in the tau pathology propagation with the aging, thus suggesting that the major risk factor is due to the fact that it increased the pathology propagation. In conclusion, we have seen that the cellular models are not good models for us to generate tangles. The mice are able to generate tangles if we inject them with the pathological tau. We have seen that the pathology can propagate between synaptically connected neurons. We have seen that the amyloid pathology can strongly increase the pathology formation after injection, and it propagates inside the brain more intensely when there is a presence of amyloid peptide. Yes. Thus, I want to thank the members of my team that generated these nice results. And I thank you for your attention, and I hope that you have questions, of course, about the topics. I hope that it was interesting for you. That was not so.
Did
follow?
everybody actually understand?
You have understood, I hope. Yes? Yes?
Okay, so you must have some question. No, okay. Sir, go ahead.
Okay.
I mean,
Okay. Yeah. There's there are a lot of factors that could affect the brain to generate Alzheimer's disease pathology. One of them is diabetes, for example. The metabolic disturbances increase the pathology. There's a model that I've shown with the prion-like mechanism. If we give the mice a high-fat diet, for example, it increases a lot the pathology propagation. It's a proof of concept that the diet is changing also the taupathology propagation. It's a link with the previous talk. There are other factors, of course, that there are polymorphism also, that there are variants in the genes that generates more risk. It increases the risk. It's not because you have the variants that you generate AD affections, but it increases the risk. It's a multifactorial disease. Thus, it's very difficult to generate a model because we do not know exactly the origin of the disease. That's why it was so difficult to generate a model. It takes decades to generate one model that is relevant for AD. And thus now we wonder if it's acting exactly like a prion. Thus, if you know, probably prion can be an infectious agent. There are grafts that gives the prion disease. And thus we wonder if it's not the case for Alzheimer's disease also, because in the blood, for example, you can detect phosphorylated tau. And thus we can wonder if the blood transfusion can give the disease also. Thus we need to be very careful about that. Thus there are a lot of studies about this type of propagation, the infection of the disease, and it's true that probably it occurs in humans. Because it was shown that some patients were treated with growth hormones. In the past, it was isolated from the bodies of humans, the brain of humans. And it was surprising because very early, at around 40 years old, they have a Alzheimer's disease. And that's why we were thinking about the prion-like mechanism. And thus, these people treated with growth hormones generate amyloid plaques, tangles, all the lesions found in Alzheimer's disease. And it's important to be careful about the human samples that we can use in all the grafts or things like that that could contain pathological tau. Yeah, and it's like the prion, it's resistant to high temperature. Thus you do not remove tau pathology, pathological tau, if you eat at 100 degrees. It remains on the tools, on everything.
wow, this is scary.
Yes, yes. yeah, but we were very surprised about that, because it's true that 10 years ago, never, all the people were not thinking that it was like this. It's very surprising for us.
So mainly we're all created not equal.
Yeah.
Gene-wise, we could be more resistant or not. Depending on our lifestyle, on our diet, we could accelerate or give more
risk.
Yeah.
Yes. Yeah, exactly. That's for example, we have some alleles in apolipoprotein. And for example, people having the LL2 are protected against AD. And it's the opposite with the LL4. They increase the risk to develop AD. Thus, probably, some people are more at risk due to the genes that they have, simply. Yeah, and the
so
diet.
what would be your advice then for some really stressed people that would like to prevent?
It's a good question because there are a lot of things that need to
so maybe
be
you
done,
have to tackle everything at the same time.
Yeah. I will say that first we need to live in a good way and not. Yes. Okay, for example, yes, of course, the sport, the diet are important, but for the
So
genes,
not
we
too
cannot
many
do.
parties, right?
Oh, yeah. For the alcohol, I don't know, honestly. There are studies about the alcohol effect, but it's not clear. But the caffeine is very good in the opposite. The low quantities of caffeine are very good.
Okay.
Thus, you can take some coffee or it's not dangerous.
I see there's a lot of happy faces. Now.
Yes.
It's like,
For the
yes.
exams and during the exams period, you can take coffee. It's not dangerous. But of course, not at high level, but the coffee is good. But alcohol, it depends, of course, if it generates, again, metabolic disturbances, it will accelerate Alzheimer's disease.
Moderation, right?
Moderation. But
you
it's
can.
important to have a good life too. Huh?
Okay, ladies and gentlemen, do we have one last question for Professor Leroy?
Yeah.
Sir?
Thank you for your presentation. I just have a little question. Do your findings give you some clues to look for a treatment for AD of the other two pathologies?
Okay. I repeat the question. Can give the advice for the treatments? Is the question, huh? It's a good question for the treatments. There are no curative treatments now for the AD. There are some treatments to I will say to inhibit the degradation of acetylcholine. In fact, it's acetylcholinesterase. It reduces the speed of the disease, the progression of the disease, but it's not curing the disease. There are other treatments that are now in phase 3. This is treatments with antibodies against tau or amyloid. Those with amyloid, the first one that was generated were not good at all. They give encephalitis and people are dying, in fact, due to the treatment, so that it was directly stopped. The second generation of antibodies are better for that and probably giving some results, but we need to have 5 or 10 years to be sure that about the effect of the treatments. It takes time, of course, in Alzheimer's disease to check the effect of treatments. There are also other treatments with cRNAs now. The cRNAs is short sequence of RNA that silence some proteins. It could be also a good tool to treat the patients. But the problem is the diagnostic of the disease, because most of the time the diagnosis is late during the evolution of the disease, and it's too late because the neurons are already dead, in fact, and thus we cannot rescue neurons that are dead. Thus, there are a lot of work to do to have good tools to diagnose early the disease. Okay. Yeah?
Yes, so. Last question?
Yes, this is the treatment with the antibodies. Thus for amyloid, the first generation were not good at all to treat the disease, but the second generation is better. Indeed, they are able to reduce the amount of amyloid in the brain, but also in the blood. In the brain, it's particularly important because we need to reduce the amyloid plaques. We have seen that the amyloid plaques accelerates the formation of tauopathogy, and is the tauopathogy responsible of the cognitive deficits. Thus, probably if you treat for amyloid, you will reduce again the progression of the disease. In this case, yes.
Thank you so much, ladies and gentlemen. Professor Karelle